I'm Jeff Rasmussen, and I'm still on my emotional webinar high from Wednesday's DNA webinar, where that light bulb finally started to turn on for me in regards to the chromosome browser and triangulation. And from your hundreds of comments afterwards, it sounds like it had the same effect for all of you. How fun to learn together. And how fun to have Mike Mansfield back with us today for another DNA webinar. This time, Introduction to DNA Testing in Genealogy and Family History. Mike gave this class for us on our most recent Legacy Genealogy cruise, and I'm happy to have him here with us to share his knowledge and expertise. And of course, I'm happy to have all of you with us today. More than 1,900 of you from these 30 countries have registered for today's live webinar. Thanks, everyone, for sharing part of your day with us. And after the main presentation, stick around for these door prizes. First, for one of our webinar subscribers, I'll have the Genealogist Google Toolbox book, the printed edition. This is authored by one of our webinar speakers, Lisa Louise Cook. Next, I'll have the 5-class DNA webinar course, Foundations in DNA by Blaine Bettinger. Then, as always, I'll have a copy of our Legacy Family Tree software for a lucky viewer. And finally, I'll have a free month's webinar membership. And now I'm happy to welcome back Mike Mansfield to the show. Mike is Director of Content Operations at MyHeritage since 2013. And in this role, he's responsible for defining the company's strategy for growing its collection of now 8.9 billion historical records and supervising all operations of content acquisition. Last time Mike was with us, just over a month ago, MyHeritage was at 8.4 billion. So he's done something well this past month and added another 500 million records since then. So well done, Mike. Previously, Mike held a senior product manager role at Family Search. His professional career has been heavily focused in electronic publishing, search and retrieval, and content acquisition and strategy. After completing his BS in computer science at Brigham Young University in 1994, he worked for Folio Corporation, a Provo, Utah-based technology company which developed cutting-edge CD-ROM publication and search technology. Mike joined Ancestry in 1999 and held key roles in its development of the search engine and publication platform still in use today. As the Senior Director of Search and Content, he led the team that created the record hinting system, which helped to revolutionize the way in which users interact with online genealogical records. He continued to develop his expertise in his roles in Family Search and MyHeritage. Well, please put together your virtual hands and let's give Mike Mansfield a nice warm webinar welcome. Mike, how are you? And welcome to the show.
It's great. Thank you, Jeff. Yeah, we had a wonderful last couple of weeks in the content group, published almost 350 million new records. So
wow, is that right?
Wonderful.
Yeah, I have to go on every day. Well, like I went on today just to check and make sure I had the number right, and yeah, sure enough. Okay, so Mike, your screen looks great. Just move that cursor off to the side, and all is well. Thanks again, Mike. It's all yours.
Thank you. All right, we're going to be talking about DNA, some introductory topics today, and here's a brief outline of how we'll go through this session. We'll do a quick biology refresher. I feel like this is an important subject to understand how inheritance works and some of the basic biological processes and structures that are in play. After that, then we'll look at the various types of DNA tests that are available in the market today, really across a fairly broad spectrum of companies and services. We'll also look at the inheritance patterns that each of those can help us discover ancestors through, or other relatives, and then strengths and weaknesses of these as well. And then finally, we'll look at some examples, some applications, and some focus studies, and I'll also be showing some demos of some of the services we have on MyHeritage with autosomal DNA in particular. All right, jumping into some biology. So here's a really interesting article written in 2013. The approximate number of cells in a human body is estimated around 37 trillion, a very large number of cells in our body. If we look at a generalized cell structure, most of the cells in our body are shaped somewhat like this. They have a nucleus where the DNA is housed, and we'll be looking more at those DNA structures in just a moment. And there's other organs within a cell that are important in the work that we do as genealogists or genetic genealogists, particularly the mitochondria that we'll talk about when we're talking about the MT or mitochondrial DNA tests. So as I said, most cells look like this, generalized cell picture, but there are some notable exceptions. One exception, for example, are red blood cells. So red blood cells do not have a nucleus, and therefore they don't carry any DNA. So red blood cells are a specialized type of cell that are highly optimized for the transmission of oxygen and other nutrients through the vascular system. So you might ask yourself, how are some of these forensic tests that we hear about using red blood cells for DNA analysis? And what they're really going for in those tests are using the white blood cells that is in all of our blood that does contain a nucleus and therefore DNA material. As we'll talk in later, the various tests in our industry, in the genealogy industry, are a swab or a spit test. So there's really no blood involved in the test that you'll do really in the genealogy domain. When we look at the human genome, it's composed of 23 chromosomes or chromosome pairs. So you often see these maybe counted where we have 46 chromosomes. That's a true statement, but they come in pairs. And so we also can say we have 23 chromosomes or 23 pairs of chromosomes. So the first 22 chromosomes are what are called autosomal chromosomes, and they're normally pictured in a diagram like this called a karyotype or a karyogram, where they're ordered roughly in size or the length of the individual chromosomes. So we have 1 through 22, with 22 being the smallest of the autosomal chromosomes. And so the chromosomes are generally numbered this way in sort of the standard fashion that's been adopted by researchers and scientists. The X and Y chromosome, as we're probably aware, are the chromosomes that determine gender. So a female will have 2 X chromosomes, and a male will have an X and a Y. So to spin a little bit of an analogy, so chromosomes are like shoes, and that is they come in pairs. And this is an important thing to understand as we go through some of the other biological processes. So when we think about chromosomes, we're often talking about a pair with both the 2 large strands of DNA that we'll talk about here in just a moment. So if we look at a human, for example, we have, as I said, 46 chromosomes, or 23 pairs. A Tibetan fox has 36. An earthworm also has 36. Rabbits have 44 chromosomes. We're kind of right in the middle then at 46. Elephants have 56. Silkworms also have 56. Hedgehogs have 90 chromosomes. And the kingfisher has an amazing 132 chromosomes. So the number of chromosomes in a particular species, as you can tell, doesn't necessarily indicate any sort of higher level of development or intelligence. It's just based mostly on how these species developed and evolved over time. When we look at a chromosome, so on the left you'll see a chromosome in broadly an X structure. In the middle there, I don't have in the diagram, but in the middle where the X crosses is what's called the centromere, where the 2 sides of this pair of this chromosome, these DNA molecules sometimes connect, or often are connected together. And so the DNA is this double helix molecule which wraps around these proteins called histones, which forms sort of a scaffolding that helps organize and keeps this very long strand of DNA in an organized way. And then those histones are in turn sort of wrapped up into these supercoils. So when we're looking at a picture or a diagram of a DNA or of a chromosome, understand that there's a lot of genetic material in there. Some of these molecules are extremely long structures. So let's look a little bit more at DNA, or deoxyribonucleic acid. That's a long word. There's basically four what we call nucleotides, and these are adenine, thymine, cytosine, and guanine. And these are the main sort of coding elements in DNA that helps determine our genetic makeup. And so if we look at the chemical structure of these nucleotides, the main thing I'm trying to point out here is that adenine and thymine will bond with just 2 hydrogen bonds, where cytosine and guanine bond with 3. That means that adenine and thymine only ever bond with each other, and cytosine and guanine only ever bond with each other. So there's sort of this compatibility between these 4 different nucleotides. So scientists and researchers have determined that these 4 nucleotides in a strand of DNA, when it's unzipped into what's called RNA for the creation of proteins and other amino acids that we'll discuss in just a moment, that they've discovered that these 3, that the nucleotides are in triplets. So here I'm showing an example of a adenine, thymine, and guanine triplet. And here's a nice chart where we can decode this. So let's look at another example here of a GTC. And the way this chart works is I'll pick the G as the first ring of this wheel, and then the T as the next possible choice off of the G, and then finally I'll get to C. And so for this triplet of GTC, this is the code for this amino acid called valine. And so there are about 20 common amino acids that are used in human biology. And so valine is over here on the right, and it has a basic chemical structure. And these amino acids are the building blocks of proteins. And proteins, again, to spin another analogy, are, in my mind, kind of like Legos, where proteins are then formed to build even more complex tissue structures, other elements used by the body, and these proteins can in turn be combined into even larger, more complex items as well. So let's look at genes for just a moment. So a gene is the physical and functional unit of heredity, and they're encoded in DNA. And as we said, the main coding mechanism is to encode amino acids, the building blocks of proteins. So really what genes are about is they're containing the information of how to create proteins. And in humans, genes can vary in size from just a few hundred DNA bases or DNA nucleotides to more than 2 million. So just as a really quick example, here's 2 genes. These are the 2 commonly well-known genes that indicate a risk for breast cancer. The first one, the BRCA type 1 susceptibility protein, is encoded on chromosome 17. It has just over 125,000 base pairs, coding for a protein composed of almost 42,000 amino acids. Just as an FYI, this is the particular gene that the actress Angelina Jolie was tested for and found to have. And so she's been very active in promoting breast cancer awareness and trying to raise funding for the types of research to investigate these problems. So what these 2 particular proteins do that are encoded by these genes is these are a type of cell sanitation system, if you will. They're caretaker proteins, and they often will be involved in destroying cells that are somehow damaged or cells that didn't get properly created. And so understanding from cancer, we can see that if the janitors and the sanitation systems in our cells aren't working properly, then that will certainly lead to some cancerous types of outcomes. So the genes are mostly the same in all people, but a small number of genes, less than about 1% of the total, are slightly different between you and I or you and anyone else. And these small differences contribute to each person's unique physical features. So the human genome, there's 23 pairs, again, as I mentioned. And if we look at chromosomes 1 through 22 with the X and Y, there are about 21,000 genes with approximately 3 billion base pairs encoded in those chromosomes. I'm going to talk just for a moment about meiosis and mitosis. So mitosis on the left, this is the process that our body uses all the time, every day, every hour, every minute to create new cells to regenerate old cells that have died. So mitosis is all about creating a new copy of a cell. Meiosis, on the right-hand side, is a more specialized process that's used to create gametes, or sperm and egg, which are used then to create a new individual, a new infant, a new fetus. So this is where an understanding of how some of these processes work can help us, especially as we use some of the tools around autosomal DNA I'll be showing later in the talk today. So as I said, it's a specialized type of cell division that reduces the chromosome number in half, each genetically distinct from the parent cell that gave rise to them. And it's a source of tremendous genetic diversity, and particularly those two mechanisms that we'll talk about that introduce this diversity are recombination, or also called crossing over, and then independent orientation. In this discussion of meiosis, I'm going to use a very simplified theoretical organism that just has two chromosomes, or two pairs of chromosomes. So we could say there's 4 chromosomes here, or 2 pairs of chromosomes. So let's pretend this is chromosome 1 and chromosome 2 on the right with their respective lengths. So just like in a situation with a human organism, 1 chromosome would be the chromosome that was inherited from the father, and 1 would have come from the mother. Another structure I'll be showing in this brief discussion is the centrioles and their microtubules that help this process happen. So at the beginning of this meiosis process, there's a cell called a germline cell that is the basis for which the sperm and egg will be created. So the first thing that happens is these long strands of DNA that I show here, all kind of jumbled up, condense into these more tightly bound chromosomal structures that are often drawn like this. And then it goes into a process of replicating those chromosomes. So for chromosome 1, we have a pair, right? Call it 2 long DNA molecules, and those molecules will each be replicated. So we have a copy of our chromosome 1, and those are called sister chromatids. And I'm showing here that we now have passed through this phase of making a copy of each of the chromosomes, or each pair of chromosomes, and then the nuclear membrane will start to break down. The centrioles will migrate to the opposite ends of the cell. And then another important process happens called recombination, or crossing over. And this is where these 2 copies, again here I'm showing perhaps chromosome 1, where in this case the blue would represent the chromosome strand that had been copied that I received from my father, and green here would represent the chromosome strand duplicated that I received from my mother. And then in recombination, these chromosomes will wrap tightly around each other, and they'll exchange segments of DNA. So what is happening in crossing over is we're combining 2 chromosomes we got from our 2 parents and making a brand new unique chromosome we will pass to our child. So another one of the main sources of genetic diversity is this recombination process that happens in mothers and fathers as they're generating sperm and eggs. So now we're representing that this crossing over phase has happened, and we have chromosomes that have swapped some genetic material. And then the next phase is the chromosomes line up at what's called the metaphase plate. And another step will happen that it introduces additional variation, and that is called independent orientation. So these chromosomes could line up like this, in this configuration like I'm showing. They could also line up like this, or like this, or like this final one. In the case of 2 sets of chromosomes, there are 4 different ways. And so the equation is 2 to the n, where n would be the number of chromosomes. So in this case, it's 2 raised to the 2nd power, and we'd get 4. So if we had an organism with 3 chromosomes, there would be 8 possible ways. In a human with 23 chromosomes, just from independent orientation, there are over 8 million ways that these chromosomes could be oriented after replication and then get split into their daughter cells, as I'll show you in just a moment. So the centrioles and these microtubules attach to the centromeres of the chromosomes, and then they pull them apart. And then the DNA sort of spreads out again, and two cells are now formed from one. And that's the first phase of meiosis, and we're still have one more phase to go. This phase is a little simpler, where the centrioles will replicate themselves in each of the two daughter cells. Those centrioles will migrate to the ends of the cell. We have another chance here for independent orientation, where the orientation of the individual strands of chromosome will end up being pulled apart. Whoops, let me go the other way. So the centrioles form these microtubules, which then pull these DNA strands into separate parts of the cell, and then those cells are split as well. So when we end meiosis, we started on the left with a germline cell, and in the case of males and sperm, we would get 4 sperm cells. In the case of a girl, a female, we would have 4 potential eggs. But actually in a female, only one of the eggs is randomly selected from this process to be preserved as an egg for future use. So as I mentioned, just from independent orientation, there's over 8.3 million ways just from independent orientation during meiosis 1 that could occur. And that's, say, on the male side. On the female side, that same thing will occur. So literally when a new fetus is formed from a sperm and an egg, that's the potential combination of over 70 trillion possibilities just from independent orientation, not even taking into account the genetic diversity that will occur from crossing over or recombination. Great, so back to our analogy about shoes. So if this is chromosome 1 from my dad, and this is my chromosome 1 from my mother, my process as a male of creating a sperm, I'll create a brand new version of the chromosome 1 and chromosome 2 that I have from my parents, and I'll pass that on to my children. When I have another child, I might produce I would almost certainly, statistically almost certain, going to produce a different chromosome than what I did for my first child. So this is what makes identical twins so special in a way, is that if you're not an identical twin, then you're almost virtually guaranteed to have a significant amount of genetic difference. The last thing we're going to talk about in this section is single nucleotide polymorphisms. And so single nucleotide polymorphism, or frequently called an SNP or SNP, are the most common type of genetic variation among people. And each SNP, or SNP, represents a difference in a single DNA building block or a single DNA nucleotide. For example, an SNP, or single nucleotide polymorphism, may replace the nucleotide cytosine with the nucleotide thiamine in a certain stretch of DNA. And that's what the diagram is illustrating. Here are 3 persons. We're looking in theoretically at the exact same chromosome, the exact same position. One might have an A, one might have a G, one might have a T. And these positions in the human genome, where some individuals have one nucleotide and others have a different nucleotide, are very well studied and understood by geneticists. And so these are the basis for the autosomal DNA test that we'll talk about in just a few minutes. So it's a few other facts about SNPs. So these occur throughout a person's DNA, but they mostly occur in non-coding regions. So these SNPs are generally outside of genes. There are a few SNPs that researchers have found that appear within genes, but you can imagine if things are changing within a gene, that could have a high probability to cause some sort of significant degradation in the capability of that final protein to function, almost like a cancerous situation. So most of the SNPs that we look at are in DNA that's considered unnecessary, and sometimes it's even called junk DNA. It's between the genes. They occur in relatively high frequencies, or about 1 in 300 bases are known to be a SNP. So within our 3 billion or so nucleotides, or our bases, we have about 10 million SNPs. And as I sort of alluded to earlier, most SNPs have no effect on health or development. All right, so in review, we've talked about the human genome, we've studied chromosomes briefly, we've looked at DNA, how genes work, and we've taken a little bit of a dive into meiosis and genetic recombination, and then we've also talked about single nucleotide polymorphisms. All right, so there's our biology refresher. I think we're now well tooled up to talk about how these DNA tests can impact the work that we do as genealogists and family historians. So let's dive into some of the specifics about DNA testing. So the DNA tests that you'll find in the industry and have been around for a while. So the first one that was really developed was the Y chromosome test. And so this was a test that's available to males. I'll talk about that in just a moment. And then also a mitochondrial DNA test, the X chromosome test, an autosomal, and then finally the autosomal test. We'll also look at inheritance patterns for these types of tests, and some of the strengths and weaknesses of these. Certainly won't be an exhaustive discussion, but I just want to give you sort of an understanding of when you would consider one over the other. So again, looking at, in particular, the Y DNA, as we look at the Y DNA test in particular, this is going to test the Y chromosome that only occurs in men or boys. So one interesting thing about the Y chromosome, it has a very easy to see inheritance pattern. So this is me on the left. So this is myself, Michael Mansfield. I was born in 1969. So my Y chromosome should be a near identical copy unless there was a slight mutation from my father, and that should have come from his father, and etc. So this goes up the paternal line as far as back into ancient history. So in my case, my Y chromosome should come out of southern England, for example. So the Y DNA test, only males have a Y chromosome, and fathers pass their Y chromosome to their sons, but not to their daughters, because daughters do not have a Y chromosome. It mutates relatively slowly, which is good in a way, because there's a very good chance that if you're a male, you'll have an almost identical copy of the Y chromosome of your father, and even your grandfather and great-grandfather. So that's potentially useful in the work that we do. And then the Y DNA tests are used to examine primarily a test taker's paternal line. And these tests are most useful for determining whether two people are not paternally related. People are or aren't paternally related. So even if you're a female, though, you can certainly think about, perhaps you have a brother who would have the Y DNA of your father, and so you can often substitute Y DNA from other members of your family. Even thinking of a grandfather who may not have had any surviving male descendants, you could go up another generation to his father and see if there's other males that you could test that would have descended from that great-grandfather to find effective Y DNA that would have been available if your grandfather had had any surviving male offspring. And then Y DNA tests are also used to determine the test taker's what's called a haplogroup. Haplogroups are these kind of deep ethnicity, sort of a mix between ethnography and ancient demography, that talk about where historically these demographers think your particular Y DNA developed and migrated from. Here's a more in-depth picture. So for example, I'm a G haplogroup, and you see G there says that I'm anciently kind of coming out of the Middle East, almost from the area of Turkey or northern Iran. And all these haplogroups are subdivided into many subsections. So I'm actually a G2A, which makes me related to this ancient mummy that they found and tested. Richard III of England. Joseph Stalin is also a G2A, as is Larry Bird. I'll take him over Stalin. And the actor Jake Gyllenhaal. Let's now prepare to talk about mitochondrial DNA testing. So the mitochondria are structures within a cell that specialize in energy conversion that the cell needs to do its function. So mitochondria are special because they have their own DNA that's outside of the nucleus, and they replicate themselves. The mitochondrial DNA has been studied to an amazing extent by scientists, and they know quite a bit about it. It has 16,569 base pairs with 37 genes. So the mitochondrial DNA is passed strictly from mothers to her children. So as a male, I certainly have mitochondrial DNA in my cells that comes exclusively from my mother, and that would have come from her mother's mother, etc. So kind of the opposite of what we saw with the Y-DNA test is the mitochondrial DNA will pass down the maternal line. And if my tree is correct, that for me should come out of Norway. So let's look a little bit more at the mitochondrial DNA. So it's a circular piece of DNA located within the mitochondria of each cell. Only mothers pass on their mtDNA to their children, in this case both the sons and daughters. And the results of mtDNA can also be used to determine a haplogroup, much like what we saw with the Y-DNA. mtDNA mutates extremely slowly, even more slowly than the Y-DNA. And it has limited use for finding genetic cousins, but it is useful certainly for determining if two people are maternally related or not. So here's an example of what the haplogroups for the mtDNA look like. I happen to belong to the T haplogroup for my mitochondrial DNA. All right, another type of test, and these are fairly rare, that you'll see an X test. So when you think about the types of chromosomes that we have, they're certainly down here in our sex chromosomes. So a female will have 2X and a male will have 2Y. So the X chromosome is inherited so as a male, I get my 1X chromosome exclusively from my mother. And so for a male, it has a particular inheritance pattern, as I'm showing here by the colored boxes. If I was a female, so let's pretend I was Michelle, I would have 2X chromosomes, one I would have received from my father and one I would have received from my mother. And so we can use this type of inheritance pattern to understand what an X DNA match and test can help us do. So as I sort of illustrated, it's inherited from a relatively small set of ancestors, certainly more though than a Y or mtDNA test. It can be useful for hunting for genetic cousins. The last thing I would say about the xDNA test is it's pretty uncommon to find people that have participated in an xDNA test. So the pool of test takers that have done this is certainly very, very small. Of the tests that I've talked about, and we'll talk about, this is by far the least common or most rare form of test. Now let's talk about sort of the big boy of the genetic genealogy testing, and that's the autosomal DNA test. So when we look at our karyotype again, we have our 23 pairs of chromosomes, and if we exclude the sex chromosomes, or the X or the Y chromosomes, the X and the Y chromosome, we get our autosomal chromosomes 1 through 22. And if we look at the inheritance pattern for an autosomal inheritance, you'll see that it's everybody. So everyone in my tree potentially can contribute autosomal DNA to me. However, an important thing to know as we look at the autosomal test in more detail is there is a notion called genetic dilution. So the second generation, so this refers to my parents, for example, where I get half of my DNA from my mother and half from my father. This also means, conversely, that half of my father's DNA I do not get, and half of my mother's DNA I do not get. If we go up to my grandparents, or the next generation, where I have 4 direct ancestors, I'll get approximately 25 of each of their DNA. But again, for my grandfather on my dad's side, or my dad's dad, I would not get approximately 75% of his DNA. And so this genetic dilution continues down this predictable chart. And so by the 7th generation, where we have 64 ancestors, we're down to just about 1.5% of their autosomal DNA likely making it down to me as a 7th generation descendant. And by the 8th generation, we're even under 1%. So what this means in reality is a theoretical situation like this, where at my 6th generation, that's the second to the right column there, I might have 3 or 4 ancestors at that level for which I have no autosomal DNA in me at all. It's been effectively diluted out. At the 7th generation, going back one more step, it's statistically probable or almost certain that I'll have somewhere around 12 to maybe as many as 16 ancestors at that level for whom I have no autosomal DNA. So Autosomal DNA, and the way it splits, as we looked at in meiosis, is kind of like a big genetic soup. And so in the meiosis process that we looked at, effectively, as a father to my son, I'm only going to give him half of my genetic soup. I'm going to essentially take what I have, cut it into two containers, and throw one container away and pass the other container to my son. Which means if there's a very infrequent ingredient in my genetic soup, like I have a small degree of German, there's a very good probability that that small degree of German might get tossed out when I split my DNA into two halves, one half to give to my son and the other half he would receive from his mother. Okay, so autosomal DNA, again, it refers to the 22 pairs of chromosomes called autosomes. A child inherits 50% from their father and 50% from their mother, and testing is done by analyzing hundreds of thousands of these SNPs, these single nucleotide polymorphisms. And ATDNA tests are very useful to hunt for genetic cousins and common ancestors. And this is done by estimating how many generations have passed and by looking at other traits that we have. And I'll show these some examples to you in a few moments. Another important thing to understand is not all genealogical cousins will share autosomal DNA. So I'm the youngest of 6 boys. I don't have any sisters. And even between my brothers, we have different pieces of our ancestors' autosomal DNA. So as I sort of alluded to, I may have very little German left in me, but one of my other brothers might have gotten a stronger dose of German when his particular autosomal DNA soup was created, so to speak. And then if you look at cousins further away from you, first cousins, second cousins, third cousins, by the time you get out to about fifth and sixth cousins, it's more and more common that you will share a very little autosomal DNA. So autosomal DNA is certainly used to estimate ethnicity or to create ethnicity estimates. And it's extremely useful for determining whether two people share a recent common ancestor, and I'll show this to you. Let's first talk about ethnicity estimates. So these are percentages of a test taker's DNA determined by the company and the company's algorithms to be associated with a particular continent, region, country, or ethnic group. And I'd like to point this out. It's not called a report. It is called an estimate because it is that. It's an estimate. And ethnicity prediction is still a relatively young and developing science. I think it's certainly going to get better, and the results today are still quite impressive, but not without some drawbacks and concerns. So how does it work? So the test taker's DNA is compared to one or more reference populations. And the individual segments of the test taker's DNA are assigned to the reference population that they most closely match. So assuming that this assumption is with this process that the DNA most likely came from that population at some recent point in time. Generally in the last, say, 200 to 300 years is kind of the resolution that these tests will be examining. So reference populations are an important aspect for these ethnicity estimates, and these are constructed through panels or groups of users that are identified and asked to take just a standard test like we all take, that they're not special in any ways. It's just that the individuals are identified for particular traits and interesting aspects of their either where they live, where they born, who their parents were, who their grandparents were, etc. And each of the major DNA testing companies have compiled their own panels, which they are used respectively for their own ethnicity estimations. So you can often find at the various companies statements like this. So here's a statement from Family Tree DNA that talks about how their panel is composed of numerous individuals from 24 different population clusters. And the statement we have on MyHeritage talks about how our current reference panels comes from over a selection of 5,000 users who we handpicked by looking at their trees for very strong genetic consistency, and we set kits out to almost every country on the earth to collect these samples. So I want to show you an example of what these ethnicity estimates, or several examples of what these ethnicity estimates look like. And before we do that, I want to sort of show you what a perfect result for me would look like, would match something like what my tree would say. So if I go out to my 7th generation in my tree, in my GEDCOM file, or my tree in Legacy, my tree on MyHeritage, here's what I would see at my 7th generation. So I have 64 direct ancestors at that level. 12 of them come from Denmark, 10 from Norway, 10 from Sweden, 24 from England, 6 from Germany or Prussian, and then 2 from Germany or Prussia, and 6 from colonial America. So what this means is I'm about 50%, exactly 50% Scandinavian, if my tree is correct, just under 40% English, a small percent German, and then about just under 10% probably unknown European. So these are these 6 colonial Americans that I've never been able to get across the pond. They could be Dutch, they could be German, they could be English. It's possible they could even be early Jewish immigrants that were known to immigrate in some of those early Dutch colonies. And the births for my 7th generation occurred between 1764 and 1819. So all 64 of those people were born in that date range. So here's what an ethnicity estimate looks like at MyHeritage. You see at the left, we're saying I have a fairly strong Scandinavian, 35%, lower than one might think, a much stronger North and West European percentage, and a smaller English. But I'll talk about how we can look at some of these results in just a moment and interpret these, I think, with a little bit more rational thought. And so here's a report from Ancestry, a very similar type of report. In just a few slides, I'll show you a table of these different ethnicity estimates. You can see them side by side. But as you can see, the major testing companies provide a very similar sort of map-based user interface where I can see the various geographies and localities of my ethnicity, 23andMe, and finally living DNA as well. So this is sort of the chart that I wanted to get to. So in the left column, again, I've represented what my tree would indicate my ethnicity would be. And then next to that, I have MyHeritage, Ancestry, Family Tree DNA, 23andMe, and Living DNA. And what you'll notice is there are certainly some significant differences. And this is common. I often speak to people at conferences and shows who have very similar results like this. And what they often do is they come up to me with their hair sort of on fire, and they want to know, how could we have bungled this up so badly, or whatever. So for example, if we take my case here, let's talk about this in a little bit more detail. And the first thing I would do with a person like this is I'll help them get their fire out, and then we can have some nice conversation. So when I look at my ethnicity estimate, it's got a much stronger North and West European component than I would have thought from my tree. But if you look at the areas that we're drawing here, 12 of my ancestors at the 7th generation come from Denmark. And so Denmark is included. Large portions of it, or large percentages of that population, are likely included in this North and West European category, even though Denmark today is considered one of the Scandinavian countries. In fact, if we look closer at Denmark in Copenhagen, so just across a narrow seaway is Sweden, over there, the city called Malmo. In fact, it's so close that there's even a bridge today. It's a very long bridge of about 5 miles, but you can actually drive from Denmark to Sweden and vice versa today. So very close geography. So what I'm trying to suggest is even some of my Swedes and some of my Swedish family did live in Malmo could also be included in this North and West European ethnicity estimate. Another nice feature we have on MyHeritage, if I go to my tree, I could show you how I can produce a heat map that shows me where the 7th generation ancestors were born and lived and died. And here's the map that I get. And you'll see just north of Germany there on the Jutland Peninsula, which is Denmark, and some of the islands of Denmark, I have quite a few results. I also have a lot of ancestors from southern England. So going back to my ethnicity report, you can see how that circle that covers all of Western Europe can pick up some of my English ancestors and likely many of my Danish and even southern Swedish ancestors. And then finally, it's always interesting to look at the history. Until 1660, this southern portion of Sweden was actually part of Denmark. So, what is Sweden? What is Denmark? It really depends when your ancestor was there and some of their own ancestors. All right, so the core concepts with ethnicity estimates. As an ethnicity estimate represents which persons of a test taker's DNA match one or more reference populations. And a reference population is a collection of DNA samples representing a particular geographic population at some fairly recent point in time. And each of the testing companies offer an ethnicity estimate with their autosomal DNA tests. And there's also third-party tools such as GEDmatch, which I don't have time to talk about in this session, offer additional ethnicity calculators. I have seen there are other sessions at legacy webinars that do go into some of these other tools such as GEDmatch, and I'd refer you to those where you'd be able to learn about how to take your data, say, from a service like MyHeritage and transfer it to GEDmatch and look at other ethnicity estimates as well. And ethnicity estimates can provide useful information to genealogists as long as we bear in mind the limitations, some of which I spoke about. And others here, although based on sophisticated algorithms and data collection schemes, they are still estimates. And some ethnicities are genetically very similar to and hard to distinguish from other ethnicities, so mistakes may, I would almost even say will, happen. And it is a good practice to treat your ethnicity estimate results with caution. Each testing company and tool will produce slightly or even significantly different results, as you saw from the table that I showed you from my own tests. So is it bad science? And this is a question that we sometimes get. And I would say the science is good and interesting, but it's still new and developing. And as we get more and more test takers, I think it's going to improve significantly over the coming years and decades. Is it clever marketing? It's certainly, without question, that you can't watch TV much anymore, at least here in the US, and not see a commercial that's advertising an autosomal DNA test. And those tests are often focused exclusively on ethnicity estimates. So to me, I'm actually okay with this because I view this as a type of gateway drug where definition of this is a habit-forming drug that, while not itself addictive, may lead to the use of other addictive drugs. So what I've found, and I think many people would agree, that we have family members who maybe haven't had an interest in genealogy and family history, but they are interested in taking a DNA test. And that often turns them into budding or novice or new genealogists because we've sparked an interest in them. And that's a great thing for us as genealogists. The more people we have come into our hobby that we love and participate with us greatly improves what we can do, especially with DNA, as I'll show you in some subsequent slides. So let's look next at some applications of using autosomal DNA in our research. The first one that I want to talk about is how we can use it for fishing for cousins and ancestors, and also include some demonstration of our website in this section. So the first thing I want to illustrate is really focused genealogical questions can be answered with DNA. So there's one particular article I want to reference by Dr. Thomas Jones, and I just noticed the other day that I believe he gave a talk in the Legacy webinar series about this specific article that I'm going to be providing a brief summary of. Dr. Jones has been very kind to allow me to give a summary of his article here, and so I certainly have his blessing and approval to show this to you. But I would refer you definitely to his article in the National Genealogical Society Quarterly and the other Legacy webinar that I think you'll find by Dr. Jones, where he talks about this case in more detail. So what Dr. Jones had is, for many years, he had been trying to he had a brick wall in his family history, this Nathaniel Greenfield at the top. He was unable to determine who his parents were. So his question was, who are the parents of this Nathaniel Greenfield? And after many years of looking, he found only 3 documents or historical records that mention Nathaniel Greenfield. That's the 1830 census, an army enlistment record, as well as an early U.S. military court-martial. However, none of these identified anyone related to Nathaniel Greenfield. He certainly had some information about where Nathaniel lived. I think this is upstate New York. So the process that Dr. Jones talks about is he decided to really test out what autosomal DNA testing could do for him in solving this brick wall. So he supplied enough autosomal DNA testing kits to Nathaniel's descendants, who he had strong evidence that they were descendants, in order to get enough possible links to other fifth cousins. And then he identified a candidate couple for Nathaniel Greenfield's parents and found their living descendants. So he did a bunch of descendant research to find potential cousins of himself and other cousins who he knew to be related to Nathaniel Greenfield. And then he supplied autosomal DNA tests to these potential cousins or potential descendants of Nathaniel's family that he was trying to prove or disprove. And then he's going to compare the test results of both pools. So the family that he found was this Thomas Greenfield and Mary Walters, living in the right place in New York about the right time frame with some other children, Luther, Caleb, and Lovila. And so his question is Nathaniel Greenfield related to this family, and if so, how? And so he did this additional descendancy research to find these living individuals at the bottom and had them test with an autosomal DNA test. And his analysis summary was that the descendants of Thomas Greenfield and the descendants of Nathaniel Greenfield, they have DNA matches. They share autosomal DNA. And so in his analysis, he determined that 5 possible relationships could exist between Nathaniel and Thomas. They could be first cousins, uncle and nephew, full brothers, half-brothers, or father and son. And after doing additional analysis with the DNA and looking at the various ways that the DNA matches were indicating how closely these people were related or distantly related to each other, he concluded that Nathaniel was Thomas's son. And Dr. Jones is one of the thought leaders in our industry about doing very rigorous genealogical studies and having excellent evidence and analysis. And so when he says that he's made this conclusion, it comes with quite a statement of authority. All right, so with that, let me jump over to MyHeritage, and I'm going to show some examples of what these autosomal DNA tests matches look like. So here's MyHeritage. This is my sort of portal or home screen. I'm going to go to DNA, and this is where I can see my ethnicity estimate. I won't spend much time on that now, and I can look at DNA matches. And I also want to show you a few things about our chromosome browser. So let's go to DNA matches. So it shows, here's me. It's going to give me a brief summary, I think, of my ethnicity estimate that I had shown you earlier in the talk. Great, so I have over 2,655 matches. The first match, right now, this is being sorted by the people with the most common DNA, and I can sort this in different ways. So my mother comes up as my most shared DNA match, as I'd expect. I share about 50% of her DNA. And then I have 4 of my 5 brothers have also tested, and they show up with about 38, 36% DNA. Below this, this set of my brothers, I also see some other people that I recognize. This is my aunt, this Janine Griffiths, another aunt, Evelyn Wallace. So I see some people I recognize. I'm not too interested in looking at them as I already know how I'm related to them. And then I start to see people that I don't recognize or don't see how I'm related. And so let me go to an example. I want to show you an example of a match that I have to a gentleman in Norway. So I get some information about this individual. He's in his 60 years old or older, and probably under 70. It gives me some great information here that he has a very large tree on MyHeritage, much larger than mine even. So this is potentially a tree that I could find a lot of new relatives in if I can understand how I'm related to this individual. So when I review his match, I can see information about the match quality. This is sort of beyond the scope of this discussion. I would refer you to other sessions, definitely in the Legacy webinar series, where they'll talk about some of these measurements, such as centimorgans, and how you can use those in your analysis. So I'm related to this individual somehow, and I can also see that I have a bunch of other smart matches. So these are smart matches or matches between my tree and his tree. So this is giving me some good evidence that I have some common ancestors are already in my tree with his. It also gives me some information about common surnames. And in Norway, we all have a lot of the same surnames, Andersons, Christiansens, those type of things. And then as I go down the list, I see matches between this gentleman in Norway and other matches that we share. So my mother shares a match with him, as do some of my brothers and my aunts and uncles that I mentioned. You'll notice here that one of my brothers, my brother John, does not share a match with this man, and I'll show you what that looks like in the chromosome browser in just a moment and why we would expect that to happen. So I come down here near the bottom of the report and I see the pedigree chart. I have two tabs. One, I can see my chart out to 7 generations, and I can see his chart. And so this gives me the ability to kind of look through his chart and see if I see any good, strong candidates. And the one that immediately jumps out at me is this surname here, this Andresen. This is a surname that I'm familiar with. It's the surname that my mother identifies with as far as her Norwegian ancestors. And when I look at this pop-up, it's also showing me that they're from the right county and even parishes that I recognize in the very far north of Norway. So there's very likely that I'm related either through this man himself or perhaps through his parents. So traditionally what you would do is you would then look at this man's tree and look at your tree, find where these individuals match, and then you can start incorporating or doing analysis on what data he has that might help you in your own research. Another thing that I can do is I'm going to come back up to the top to the DNA menu, and I'm going to show you what we can do with the chromosome browser with this exact same case. So this chromosome browser, we released it just over a month ago, or just less than a month ago now at RootsTech. This is our one-to-many chromosome browser, and this has been an amazing tool that I hope you get as excited as I think Jeff and I are about this. So let's, for example, put in my mother, and let's just do a quick compare. So this is going to be sort of a one-to-one. I'm just comparing me and my mother. So as I expect, I have a large degree of shared DNA with my mother. Let's take off my mother, and I want to put in my 4 brothers that have tested. I've got to get my 5th brother to complete his test. So I have Scott, John, Steve, and Mark. And I've made a group of 5 people then. So these are 5 brothers, myself and my 4 older brothers. And here's what I see. So my analysis between myself and Scott is in red. My analysis with myself and John is in this sort of burnt orange color. Yellow is Steve, and then green is Mark. And these triangulated segments, what this means is these are segments that this group of 5 people all share together. And what you'll notice as a group of brothers, we don't share the exact same autosomal DNA, and that goes back to what we talked about in meiosis and how the sperm and eggs are developed. And so we shouldn't be too surprised by this. And so I want to go back and show you a more practical application of what we can do now with the chromosome browser. So as I mentioned, my mother identifies fairly strongly with her Norwegian heritage. And this man that I showed you that I have a DNA match for, so let me find him again in my set. So a very good technique to use when using a chromosome browser is use someone that you understand your relationship to, you have evidence for, and then use that to triangulate with someone that you don't know. So I'm going to put in this man from Norway. So if I get a triangulated match here, this is going to be very good evidence that this man is related to me through my mother's Norwegian ancestry. And sure enough, I get one triangulated match. And then the rest of this is my mother's DNA, as I showed earlier. So this is a fairly distant relative. I believe he's something like my 4th cousin once removed, or 5th cousin. So this just starts to show you some of the things you can do with this incredible one-to-many chromosome browser. Jeff was in a session just 2 days ago where he went through this with some of his family. I'd refer you to that webinar as well if you're interested. Let's finish off here then with something I've been given permission to show you. So this is software that's still in alpha at MyHeritage. So alpha is a way a software company talks about things that are still under internal development. So it's kind of like, say, it's under heavy construction. And then as users, we'll often see what's called beta software. And these are, usually some sort of limited access or even wide access, but still with the notion that it's under construction. So what I'm about to show you is alpha software. And this was something that I got permission for from my executive, and I appreciate his approval to let me share this with you. So we call this the theory of family relativity, kind of playing on Einstein's theory of general relativity. But what we're doing is we're computing possible relationship paths between DNA matches. We're trying to help automate that process that I showed to you of determining how my Norwegian match might actually fit into my tree. So we do this by building a very large graph database. So we take all of our family trees, all of our historical records, everything that we have that we know about people in MyHeritage, and we make these connections between all these individuals. And then what these theories of family relativity do is over on the left, in the lower left corner, you'll see myself. So Michael Lauren Mansfield, that's my full name with my middle name. And it shows that this theory will walk up my tree to my great-grandfather Matthew Lorenzo Mansfield, and it jumps over into another tree, goes up for 4 generations, seems to verify some information in the 1850 U.S. Census, and then it goes down several generations to one of my DNA matches, this lady whose first name starts with Wendy. So this is work that we're working on at MyHeritage to help us understand how these matches work into our tree and how we can verify this also with the chromosome browser that I showed. Another example of another theory from the Theory of Family Relativity project is, again, myself over on the left, going up my tree, going through the family search tree, going into another tree from Norway, and then into a second tree from Norway to get back down to that same person that I just showed you of my DNA match, this gentleman named Borge. So according to this theory, this is one way that I relate to him, and he's my 4th cousin once removed. Here's another example of a record. This is not me, but someone on the left named George Douglas, and I've obfuscated his last name. It goes up his tree several generations, jumps over into another tree, and then it goes through an 1860 U.S. Census, picking up a mother and her daughter, and then uses that daughter relationship to go into another tree on the right, and then down to a match that this George Douglas on the left had with this James Wilbur on the right. And then one of the more complex examples I would show you is something like this. So this is one of my own theories where I'm on the left. It goes through a tree from Canada, the family search family tree, and then 2 trees from Sweden, and then back into a tree from the US to reveal to me that I'm likely a 5th cousin to this match that I have. Her first name is Beverly. All right, so just quickly in the last few minutes that we have, the testing options for autosomal DNA, there's really 5, maybe 6 if you want to count the National Geographic test. The main players are here: MyHeritage, Ancestry, 23andMe, Family Tree DNA, Living DNA, as well, sort of the newest entrant into the market. Normal retail for these tests is about $75 to $100, and you'll sometimes see specials and sales where you can get them around $60 to $80. Non-DNA testing or non-autosomal DNA testing, the company you'll want to look to for that is Family Tree DNA. They're Y-DNA tests. So they have 3 different levels. They do get somewhat expensive. They have an mtDNA, 2 different options there, and then some other advanced tests. They even have some fun, what they call factoid tests. So at Family Tree DNA, you can take a test for $19 to determine if you have wet or dry earwax, if you didn't already know. So this gets sort of the gross EU award for the talk. With all these testing companies that do autosomal DNA tests, you can also download or export your raw data. So here's the top of what a very large file of 700,000 of these rows would look like. It gives me information about what's called the RSID, the chromosome, and the position. Those position and RSID numbers are numbering techniques that are used by geneticists to identify the position on the chromosome, and then the particular nucleotide on the first strand of DNA, or the allele 1, or the second strand in that chromosome, allele 2. So you can download this data, and that gives you some nice options. For some companies like Family Tree DNA, you can transfer your autosomal test to them. Same with MyHeritage. So if you've tested at one of the other providers, you can transfer to MyHeritage. Some of the other companies do not allow autosomal DNA transfers, such as Ancestry and 23andMe. And then living DNA, I just read that they're working on their transfer capability. And then if you have your autosomal DNA data, you can also transfer to some of these other third-party tools such as GEDmatch and DNA GEDCOM that you can do some other fun analysis with some of the tools that they provide. If you want to learn more, I would refer you to this really nice website, the International Society of Genetic Genealogy. There you'll find a number of resources. One set that I like is they have a nice list of blogs of specialists that write and discuss genetic genealogy. Two that I follow are the Genetic Genealogists, and the other one is Your Genetic Genealogist. The first one I mentioned there, The Genetic Genealogist, is actually written and maintained by Dr. Blaine Bettinger, who I see is actually presenting here in the webinar, series. He has a talk coming up next week and another one in May. He also has 13 other webinars already in the library that you can go look at right now. Many other DNA experts and topics are also in the webinar library right now. So just a tremendous resource here at Legacy, and Jeff and the team at Legacy should be commended for the work that they do. So in conclusion, initially I kind of wanted to say, DNA is a new tool in our toolbox, but it's really starting to be an entirely new toolbox. So we have our old techniques, our awesome, amazing, tried and true techniques as genealogists and family historians. Those don't diminish. And we have a new toolbox that we're learning about that's developing in genetic DNA. And it's exciting to see how we can combine those tools together, as I've shown you in some of the stuff that I looked at today. Some credits of some of the licensed images that I used, and now we'll take some questions. And again, my name is Mike, and here's my email address. I'd be happy to hear from you if we don't get a time to chat during the Q&A session. With that, let's go to Jeff.
Okay, Mike, I love your genetic soup concept and all of the other pictures that you use, the shoes. I mean, I'm just saying this because we got dozens and dozens of comments about how you turned biology into something that we can actually understand. So well done. Mike, I'm going to switch over here to my computer for a minute, and let's do some door prizes. And then we've got a good list of questions here to ask of you in just a minute. You'll be able to go and re-watch this webinar. I'll have it on within a couple of hours after we conclude here today. And right after I get done with that, it's spring break. We're off to the Oregon coast with the kids. Looking forward to that. Okay, let's show you that brand new in the library. This is the one on Wednesday night, so that's up there, hands-on with MyHeritage DNA. Just this morning, we released from Gina Philibert Ortega, Social History Through the Ages. So that's our bonus webinar for members that we release every Friday. And this Tuesday evening webinar from BCG, this was called as one of the finest webinars we've ever had. If you've got World War II ancestors, then we've got a whole 4-part series for you. And then Cindy Engel of cindyslist.com was also one of our recent speakers. Here's the direct link for the 44 classes. I think actually 45 classes now up in the DNA in the webinar libraries, and many of these are from Blaine Bettinger. Here's the one from Tom Jones that Mike was talking to us about, so that's up there as well. And if you want more Mike Mansfield, Mike was key to well, some big decisions in my personal life with how he taught about technologies at MyHeritage. So thanks to Mike. But he's got many good talks up there that you can view. Next Tuesday, we're going to have the next MyHeritage webinar series webinar on how DNA testing can reveal your ethnic roots, followed by the next day with Blaine Bettinger on formulating a DNA testing plan. Beginning of April, we'll have Jewish genealogy for the non-Jew, history, migration, and DNA, followed by Legacy 9 Unlocked, adding a census record in the Watch Jeff Live format. And then April 10th, we'll have genealogy for advanced users, grow your family tree online. Okay, it's time for door prizes, and I'm going to start doing more of these kind because we've got a whole inventory in at the office of just so many of these great books from our webinar presenters. So We're going to start out with the Genealogist Google Toolbox, and this is by Lisa Louise Cook. You can go and grab this up at Genealogy Gems. If you're here for the first time, then I'll let you know that if you're a webinar subscriber, then your name is always in the pool for one of these webinar door prizes. And today, this will go to Richard Feinberg. So thanks, Richard, for your membership, and congrats. Now I'm going to go on to DNA course number 1. This is the Foundations course by Blaine Bettinger. So he starts you out at the very beginning, and he really just holds your hand, and by the time you're done with number 5, you've really got a good foundation for everything else. And let me go and let you raise your virtual hands now. So if you already have a webinar subscription, then you already have access to this in the webinar library. So maybe raise your hand if you don't yet have a membership. This will also come with all of the handouts. They're $9.95 each if you want them separately, or with your webinar membership, it is included. All right, we're going to go. Let me go and find who's raising their hands. Let's go to Marta Bertgren. Marta, congrats. Good to have you here today. Thanks for clicking at just the right time. All right, now we've got Legacy Family Tree software. And we've got some DNA charts in there that are I quite like. So go ahead and yeah, raise your hand again. Maybe you don't have Legacy. Maybe you want to give this to someone else. All right, gotcha here. Cat Todd. Cat, congrats. Thanks for being here today. Now let's go on to webinar membership. Yeah, we're getting close to the 700 classes mark up there. I think we've surpassed 900 hours of continuing education up there in the library. But it's not just any 900 hours. We only get the best speakers, and you know that. Okay, $49.95 a year or monthly at 10% off with today's coupon. Let's go and find, looks like D. Kermodi. Just writing down your names here. D, congrats. Just watch for an email from me. And there's your coupon code, everyone. So DNA Intro. Yeah. DNA Intro, just use it by Monday, 10% off. Our quick guides, memberships, digital download, software, whatever we've got up there. All right, Mike.
Yeah.
Welcome back. All right.
Thank you.
I got a whole bunch of comments. Just one I'm going to read, and then let's go into questions. Sue says that ethnicity estimate explanation blew my mind. It all makes sense now, and I can't wait to look at my tree with fresh eyes. So for Sue, she's real excited to have been here today. So Mike, right when you're talking about the heat map, I had all kinds of questions come in here saying, hey, how do we find that heat map? Do you mind if either on your screen, or you can walk me through on my screen, taking me to the heat map?
Yeah, absolutely, no problem. If you want to show my screen, I can show it.
Okay, here you go, Mike.
All right, and so there's kind of two forms. Let me show you when I go to. Let me just show you the main one. So here under Family Tree, so this is where I manage, my trees on MyHeritage. And there's this, it's a feature of this larger feature set we call the pedigree map. So this fourth option down. So the pedigree map it's also a wonderful tool for looking at many other aspects of your tree and helping you get data sort of normalized and consistent. But the heat map function is certainly fun. So I'm going to zoom in here to sort of the view that I showed you of my family in Europe. So it's showing me by default some of these events that it's counting, various things. I can turn on some filters. So here's where I say, what generation I might want to limit it to if I want to include just births. And then down here at the bottom, in the lower right-hand corner, I can say heat map. And so this shows me this view of events that are occurring in my tree based on the filters that I've set. I can also do a year range filter or various things. And it's coloring here where I have ancestors that were born in this case, and other. Let's turn off other. So I have several different families in Sweden, several in Denmark, several in Norway, and way up here at the top in Norway is one of my mother's family that were fishermen way up there in the north, over here in London. I hope that answered the question.
Yep, very good. Yeah, love the heat map. Thank you so much. Let's go to Marilyn's question, who asks, who's looking for a confirmation. She says, so does that mean I wouldn't come up as a DNA match to a 6th or 7th cousin? Is it possible that she would, or possible that she wouldn't?
It's possible that you could, but it's increasingly unlikely as you get, the 6th and 7th cousins.
Yeah.
Now there are strategies you can start to think about that can help you kind of bridge that gap. So let's say you had a second cousin that you had a known genealogical relationship with, and if your second cousin has taken a test, you're 2 degrees closer to that 6th potential cousin that you're trying to determine. So whenever you're doing DNA or work with DNA, think not only about your own test, but what the tests of your siblings, your parents, grandparents if you can, nieces and nephews, cousins, second cousins. I regret that I didn't get a test from my father before he passed away a few years ago. I've tested my mother, and I'm certainly going to get my father's living brother and his 2 sisters tested as sort of a surrogate for my father's DNA. I think Jeff on Wednesday mentioned that he has tests for, I think, his dad and both of his grandfathers. Is that right?
Yeah, three of my 4 grandparents I was fortunate to get the tests of, which leads us to Brian's question. Is it possible to reconstitute DNA? So I don't have my dad's mom's DNA. Can I have it created somehow from what I do have?
Yeah, so that's a very interesting question. And sorry, Jeff, you had something?
Well, Mike, I'm getting something from your mic. Could you unplug your mic real quick and then plug it back in, and then the rest of our Q&A will be happy.
Better. Sure. Let's do it.
Thanks.
Am I back?
Yeah. Yep. Thanks, Mike.
Okay, so the question was, can you reconstitute DNA for, say a grandparent or someone like that? So this was actually spoken about at RootsTech by our CEO, where when you start to look at what you can do with triangulation, as we showed just really briefly in the talk, you can start to identify segments of DNA that you can, with a high degree of probability, assign to someone who's deceased. With enough of these types of samples, you can start to imagine, and this is certainly where research is being conducted to operationalize this, that in the coming years, I think that the answer is yes, you'll be able to start to reconstitute certainly pieces, and with enough data, potentially entire pieces of DNA. But remember, in the autosomal test we're only looking at about 700,000 of these SNPs. We're not trying to build the whole genome for these people, because we just don't have any of the data to support that. But as far as what we do for genealogy, you could say yes, we're certainly on the road towards being able to recreate and assign DNA to persons who died before testing was even available.
Cool. Can't wait for that to come. Thanks for that answer. Here's a real quick answer or easy question, but from Karen. Can I upload my Ancestry DNA test to MyHeritage, and what's the cost?
So the answer is yes. You can download your DNA from Ancestry, and you can upload it to MyHeritage, and there is no fee for that service. You'll get the ethnicity estimates as well as DNA matches. If you want to do additional work with some of the trees and investigating what other members on MyHeritage have in their trees beyond what the some of the tools that I showed you would do, then there could be a subscription service that you would want to investigate at MyHeritage. But it's certainly worthwhile to see, if you want to see what the ethnicity estimates look like and to get the free DNA matches. You can definitely get a feel for what the service could potentially help you do.
Okay, thank you so much, Mike. Now, are you familiar with the word or the concept of phasing? It's something I've heard of, I think, from Blaine Bettinger. But Donna's asking about that, wondering if phasing could be related to reconstructing someone's DNA.
Yeah, I'm going to leave that for deeper experts than myself.
Yeah, so Donna, seems like Blaine talked to us about phasing in one of the webinars in his library. Maybe go over there and maybe send Blaine or go on to the Facebook page that Mike showed us. What is the genealogist?
What his blog was?
Geneticgenealogist.com. Go there. I'd probably point to his Facebook page and ask about phasing over there. But an interesting, more in-depth question. Thanks for that. So, question from Marcia about haplogroups. She says, so if a man has a haplogroup of K, would his son also have that same haplogroup?
Yeah, the answer is absolutely. The mutation of the Y chromosome is so infrequent and so rare that even if there was a slight mutation, the son would still be considered some sort of K haplogroup. All those haplogroups are subdivided into many different what they call subclades. And so while I said I was a G2A, I'm also a much deeper sort of subdivision of that larger G2A category. So if your son was the first occurrence of that mutation, then they would subclade him into a new subsection of K, or if that makes sense.
Okay, thanks, Mike, and thanks for your question. Arlene is asking about the xDNA test and is wondering, why is it so uncommon? Is that just something, is it tech that's not developed yet? Will there be something in the future that'll take advantage of the X?
Yeah, and when you go to even family tree DNA, it's hard to even find information about the X testing and some of the capabilities that they have. There are some forums that they have that I would encourage you to look through. It just hasn't been, I think, the focus of the larger companies. Most of the largest companies are putting most of their development right now into the autosomal capabilities. Mm-hmm. You'd imagine in time, if we really kind of knock that out of the park, although there's certainly a lot of work to do to improve the tools and systems that more attention might be given by the industry to the X and some of these other tests.
All right, so we'll probably get there sometime. Let's go to Dale's question, who asks, my heritage ethnicity came up with an ethnicity group as 4.4%. And she's wondering, is that a significant percentage to consider? She says it was 2% over an ancestry. Is there a low percentage level that we need to be leery about?
Yeah, and so in the chart that I was showing you, you'll see that most of the services aren't going below maybe 2%, 1%. I personally, if it's less than 2, I think it's statistically maybe questionable how relevant it is. Okay. But still, out at the 7th generation, one ancestor is going to be, about 1.5%. So if one of your 64th 7th generation ancestors, let's say Native American, then that's potentially an important aspect you could learn from an ethnicity estimate, is if you had something that indicated Native American at that ratio, you would want to look for one of those likely seventh generations to have that type of ethnicity.
Yeah. Okay. Thank you. Let's go to this question here from Jackie. This is going back to the triangulation and the chromosome browser. And Jackie's wondering, I see many places that on your screen that had all 4 colors. Why are they not also considered triangulated as the ones that are circled? So help us understand the triangulation part of it.
Sure. So the way that triangulate, and an example I showed, I think I had myself and my four brothers, and we share a lot of autosomal DNA between the four of us, five of us actually, but it only triangulated or drew the sort of circle around the sections where we all shared the same DNA. So in an example like I used with myself, my mother, and this man from Norway, the triangulation will help me understand that this man I've never met in Norway, my mother, who I have some reasonable knowledge of her ancestry, and myself have some common ancestor between the three of us. And their triangulation group could be, it could be 3, 4, it can be as many as 8 people with the current one-to-many chromosome browser. And so you can use that information to help narrow down your search through the potential common ancestors that you might have between your tree and this other person's tree, or other persons in your same group, in your triangulation group. I hope that helped.
Thanks, Mike. I appreciate that. Now let's go to Murray here. I'm wondering why some ethnicity results, like the one shown, gives a breakdown of the different regions of Scandinavia, such as Denmark, Sweden, Norway, while others just say Scandinavian. Could he be talking about different companies there, or different results within the same?
Yeah, so different companies have different sort of regions or sub-regions that they will report. For example, I think Ancestry has both a sort of Britain, meaning Scotland, Wales, and England, and then a separate Irish or Ireland. It's really just a function of how confident these companies are in reporting ethnicities from their reference panels, how good they think the reference panels are and their algorithms are to disambiguate those. In other cases, like I showed with this broad northwestern Europe, it was even parts of northern Spain, all of France, parts of Switzerland, northwest Italy, a broad swath of countries through there. And when you think about identifying some of the genetic differences between those populations that were pretty fluid, migrating between at least say France and Germany or these other countries. It's a challenging problem for these ethnicity estimates to be able to segment those with a degree of certainty.
Okay, and speaking of this, let's go to, speaking of what I was just thinking of while you were talking, I think it's not related. Jerome has this question about, is there a way to determine the paternal versus maternal lines using the chromosome browser? And Mike, after Wednesday's webinar, I can now with authority say yes, and I'm excited that I know about that, but what can you tell us about that?
Yeah, and so Jeff's referring to the experience he had on Wednesday, which I watched that webinar just yesterday. And Jeff's experience was similar to mine, where I was able to use my mother in my triangulation. So when I get a match to this gentleman in Norway, I'm and a triangulated match, I have a great deal of evidence that he is related to me through my mother's DNA, and that my DNA that matches with this man in Norway indicates that the chromosome that I'm matching with on my mom came from my mom. And so this is also another thing to think about. So if I were to put my mother's sister or my aunt, myself, and this man from Norway in the chromosome browser, and I could show you this, I don't get a triangulated match. That's because my mother's sister doesn't have the same segment of autosomal DNA that my mother had that produced that triangulation. Okay. So just because you don't get a triangulated match, that doesn't prove that you're not related, right? Okay. It's when you get a triangulated match that's giving you evidence that you are. It doesn't really work in reverse. So definitely,
yeah, very good to keep in mind. Thanks for that clarification. That's great. Well, Mike, we've used you today. We've loved everything you've taught us here. The biology, the actual examples. It's been really fun. Before we officially say goodbye and good weekend, any final thoughts you'd like to leave us with?
No, just that the genetic DNA is it's an incredible new set of tools, like I mentioned. I know I've been spending time trying to get more expert at them, and I have ways to go.
Yeah.
And definitely encourage each of you to think about taking advantage of the resources, these educational opportunities like this, and others to really hone our skills at this. It's going to definitely change some of the work that we do as genealogists, or enhance it and augment what we have been doing. So it's exciting.
Well, great way to leave us here, Mike. Appreciate it. Okay,
Yeah,
thanks to you, and thanks to all of you, wherever and whenever you are around the world, for sharing part of your day with us. And remember, life is short. Do genealogy first. Bye, everyone. Bye, Mike. Thanks.
See ya. Goodbye.